What is gene therapy? Gene therapy is the introduction, removal, or change in genetic material in the cells of a patient to treat an inherited or developed disease. Genetic material such as a working copy of a gene is transferred into the target cell using a vector. A vector is a carrier that is genetically engineered to deliver the gene. It is often a virus, but the viral genes are removed. It may refer to the DNA molecule itself or to the carrier construct containing the DNA. Gene therapies can work by several mechanisms. It can replace, inactivate, and introduce. Replace a disease-causing gene with a healthy copy of the gene. Inactivate a disease-causing gene that is not functioning properly. And lastly, is to introduce a new or modified gene into the body to help treat a disease. The process of gene therapy includes 5 steps. The first is creating a working gene. The gene transfer therapy process begins when a working gene is created in the laboratory. The working gene is developed to contain the instructions for making a needed protein. Scientists design working genes to meet specific needs. The second is the building of a therapeutic vector. The working gene needs to be protected and directed on where to go in the body. To do so, a therapeutic vector is used and is created by modifying a naturally occurring virus without the viral DNA, and the working gene is put inside the empty shell. The third step is determining eligibility. Other than age, gender, and organ health, the therapeutic vectors are commonly made from adeno-associated viruses which are not known to make people sick. Candidates may have to be screened to ensure that they do not have immunity to the AAV because that reduces its effectiveness.
The fourth step is delivering the working gene. A single one-time infusion in appropriate clinical setting delivers large number of therapeutic vectors into the body. Once in the body, the new gene is designed to do work of the gene that is missing or isn't functioning properly, because the new working gene is not intended to become part of your DNA. The original missing mutated gene is left unchanged. The final step of any treatment would be monitoring safety and efficacy. Regular monitoring is important because it allows researchers to understand any risk and what impact the gene transfer is having. Patients in clinical trials meet with their care teams for blood tests and to discuss their medication for regimen and lifestyle to collect data as a part of the study. As with all the medications, response to gene therapy may vary.
This video is produced by Random 42 Scientific Notation. Many human diseases are caused by defective genes and improper protein functioning. Genes within our DNA provide the blueprint for the production of proteins. Specific gene defects or mutations can result in reduced or inability to produce a functional protein or production of a protein with altered function. Gene transfer using adeno-associated virus or AAV vectors can target cells to correct disease causing improper protein function. Once inside the target cell, therapeutic genes within the AAV can correct defective or missing protein function. To achieve this AAV, genetic material is replaced with a therapeutic gene to restore natural function of the disrupted proteins. A protein shell is called a capsid encases the genetic material and helps target delivery to certain cells. Once within the target cells, the capsid is shed inside the nucleus to release its vector genome. The vector genome does not integrate into the nuclear DNA but instead forms an episome. Episomes are not passed on to future generations of cells, thereby ensuring that the effect of the gene transfer is not passed on. Using the body's natural machinery, this allows stable and long-term expression of the therapeutic molecule. AAV-based vector delivery systems can be administered by intravenous drip or direct injection to target tissues. AAVs are ideal vectors for gene transfer as they are non-pathogenic and capable of transducing both dividing and quiescent cells. The different components of AAV-based gene transfer can be switched to address different therapeutic targets and diseases, and several studies and clinical trials have indicated that there is activity in different disease models. Gene transfer offers the potential of long-term and potentially curative benefit to patients with genetic or acquired diseases by correcting defective or missing protein function through a single administration. uniQure is committed to developing innovative and cost-effective single-treatment gene therapies with potentially curative results.
Gene therapy has now accomplished a huge number in modern medicine and is finding its path into clinic and in the market. In 2017, Rexelle, the first human gene therapy drug for the inherited retina dystrophy, was approved by Food and Drug Administration and entered the US market. In the same year, Kymriah, Yescarta, two cell-based gene therapies for the treatment of the acute and lymphoblastic leukemia, were also approved by FDA. Various outstanding gene and cell-based gene therapies for both rare and common genetic disorders as well as life-threatening disease are in the evaluation of the phase prior to their translation into the clinic in the near future. It is presently starting to become a commercially profitable as number of gene and cell-based gene therapy products have entered the market and the clinic. What is stem cell therapy? Stem cell therapy is the transfer of cells into a patient with the goal of improving a disease.
Stem cell therapy is any treatment that uses stem cells as the primary way of curing or reducing the severity of a disease or disorder. There are two main ways stem cells can be used. The first is as a transplant, where the desired stem cells are harvested either from the patient or a donor and refined or modified in some way before being injected or grafted into the patient. And the second one is as a target for a drug or other biologic, where the drug or biologic is intended to activate a desired response from the stem cells that already exist in the patient's tissues or organs. A stem cell or bone marrow transplant is a long and complicated process that involves 5 main stages. The first of these stages are the tests and examinations. Before a stem cell transplant can be carried out, you'll need a series of tests and examinations to ensure you're healthy enough for the procedure to be carried out. Transplants tend to be more successful in people who are in good general health despite their underlying condition. This is followed by the harvesting of the stem cells. After the test, the stem cells that will be used for the transplant will need to be removed and stored. They can be harvested from blood, which is the most common, from bone marrow, or from cord blood. The next step of the procedure is the conditioning treatment. Treatment with high doses of chemotherapy and sometimes radiotherapy will be needed before the stem cells can be transplanted to destroy existing bone marrow cells, destroy any existing cancer cells, and stop your immune system working. The process would then move on to the transplant, which is usually carried out a day or two after conditioning has finished. The stem cells will be passed slowly into your body through the central line. This process often takes around a couple of hours. The transplant won't be painful and you'll be awake throughout. Finally is the recovery process. Once the transplant is finished, you'll need to stay in hospital for a few weeks while you wait for the stem cells to settle in your bone marrow and start producing new blood cells. If donated stem cells were transplanted, you'll also usually need to take medicines that stop your immune system from working so strongly to reduce the risk of your body attacking the transplanted cells or to reduce the risk of the transplanted cells attacking other cells in your body.
This video is developed by Free Medical Education. When hearing the two words together, stem cells, most of us would think of cells residing inside the stem of plants. But in reality, stem cells are cells that divide to produce specialized other cells in order to replace the worn-out ones. and maintain the homeostasis. Think of it like a branch of tree, on which leaves, fruits, and small branches emerge. Stem cells work just as similar. Specialized cells are differentiated from its precursor stem cell to maintain the body's internal environment. Until now, two kinds of stem cells of human sources have been known: embryonic stem cells and adult stem cells. Embryonic stem cells has the ability to differentiate into any necessary type of body cells, while adult stem cells has limited differentiation of cells specific only to its tissue origin. To avoid the ethical issue in research, embryonic stem cells can be recreated by replacing the egg cell's haploid genome with adult's diploid genome, as in the case of the cloned sheep Dolly, or by introducing several iPS genes into an adult skin cell. The most common example a biology student could give of adult stem cell is that of blood cells. If we are hiking on the top of a snowy mountain, the rising demand for oxygen from our body is met as soon as our kidney responds by secreting a type of hormone into the blood. This particular hormone sends a signal to bone marrow for the differentiation of stem cells into more red blood cells to fulfill our body's need. Stem cells are also being cultured to create skin that can be grafted on burned bruises. Another example of cultured stem cell is cultured milk. The meat grown in a lab may be considerably more beneficial to our health and environment than conventional meat. In addition, plants are also possible to be created in labs by providing a specific amount of nutrients and growth factors for the undifferentiated, non-specialized stem cells to start generating shoots and roots.
In the past few years, a rather exponential advancement in clinical trials revolved around stem cell-based therapies. A case of epidermolysis bullosa manifested signs of skin recovery after treatment with keratinocyte culture of epidermal stem cells. Also, a major improvement in eyesight of patients suffering from macular degeneration was reported after transplantation of patient-derived inducted pluripotent stem cells that were inducted to differentiate into pigments epithelial cells of the retina. iPSCs are concurring the field of stem cell research with endless possibilities of treating disease using patient-owned cells. Regeneration of dental and periodontal tissues use MSCs has made its way to the clinic and soon enough will become a valid treatment. Although changes might seem daunting, stem cell research is advancing rapidly and cellular therapeutics is soon to be available. Fortunately, there are currently tremendous effort exerted globally towards setting up regulatory guidelines and standards to ensure patients safely. In the near future, stem cell-based therapy shall significantly impact the human health.
In summary, gene therapy and stem cell therapy are still in the process of their development. While doing so, both have managed to impact the medicine field. There might have been bumps and obstacles on the way, but better face them now than in the near future, where we hope to fully make use of them in changing the course of medicine.